Okay, hello everyone. So this is just a recording of the week 13 lecture for PSY4050 Quantitative Research Methods. So like in previous weeks, I'm just making a supplementary video or audio recording of the lecture that people can make use of. Okay, so let's get my slides working. There we go. So this week we're covering Factorial ANOVA. And the aims are to extend the ANOVA model that we covered in, I think, week 5, where we compare three or more group means on an independent variable. We're going to extend that to include 2 independent variables and try to explain how variability can be partitioned in a 2-way independent groups and analysis of variance, or 2-way independent groups ANOVA design. And the aim is also to enable students, enable you guys to interpret this also 2-way independent groups ANOVA, including the interaction effects that you might get. So after this week's lecture and lab class, you should be able to demonstrate an understanding of the rationale for a 2-way ANOVA, be able to construct and interpret the summary table for a 2-way ANOVA, and also be able to conduct and interpret post-op tests after a factorial ANOVA. So if you remember, when we run an ANOVA, when it was just a 1-way ANOVA, when we looked at it, if you get a significant result, you then have to do these kind of follow-up tests to see which groups are significantly different from each other. Okay, so just a quick recap. When we covered 1-way ANOVA, we were able to depict our data as shown on this particular figure. So we had a categorical variable, our independent variable, on the x-axis, on the horizontal axis, and we had a continuous dependent variable on the y-axis, or the vertical axis. So you can see here on the x-axis there are 3 groups. There are 5 data points in each group, and they are plotted on the x-axis against their scores on the y-axis. And the red bars there are the means of each of the groups, and as you can see, if you recall, I drew this in the lab classes to try to give people a visual intuition of how ANOVA works. But as you can see here, the group means differ from each other, so there's kind of quite a bit of difference between the groups, and the spread around the groups, the error around the groups, it doesn't overlap extensively. So we would likely find with this particular ANOVA, we would have a significant ANOVA result and a significant difference between at least some of the groups, probably all 3, in fact. So what we're going to do this week is increase the complexity of the design. So with a factorial design, we can increase the complexity of our statistical model by adding more independent variables. So these IVs are also referred to as factors. So a study with 1 independent variable has a single factor. It's often referred to as a 1-way design. A factorial design has 2, 3, or maybe even more independent variables. And you sometimes see these referred to as 2-way or 3-way designs, depending on how many IVs they have. So factorial ANOVA designs still have 1 continuous dependent variable, the outcome variable, thing we're measuring changes in scores on or differences in scores on, but they have more than 1 categorical independent variable. And this allows us to test more than 1 hypothesis. That's the useful thing about these. We can test more than 1 hypothesis. We can include potentially confounding variables into the design as factors, so to some extent control for them. And it also, factorial designs, they also enable us to test the effects of interactions between independent variables. This is the effect of them on the dependent variable. So the interaction between the variables may affect scores on the dependent variable in different ways, and we'll see what that means in more detail shortly. A couple of quick definitions though. Factors, as I've already said, these are independent variables, each with a number of levels, 2, 3, 4, maybe more. A factorial design of the type we're looking at is basically an experimental design which uses all combinations of levels of factors. And these are sometimes called cross factors. So our groups are kind of being divided by more than 1 independent categorical variable. Okay, a treatment, a common word for this, for treatment is the manipulation that we apply. It's a particular, and that will be a particular combination of levels of the factors. So if each independent variable has different levels, and when I'm manipulating participants in which one's going which levels, if we've got more than one, we'd refer to it as a treatment. And the combination of, say, a level from one independent variable with a level from a second independent variable, we now refer to as a cell. And in independent groups design, it's also commonly referred to as a group. So what might this all look like if we were trying to sort of visually display a factorial design with two categorical and independent variables? Well, we could put it in a table like this. So we have IV1 in the columns here, and this has three levels that I've just labelled 1, 2, and 3. And IV2 is shown in the rows, and this has two levels that I've just represented as A and B. Okay, and this would be what we might refer to as a 3x2 design. So it's got two independent variables. The first one has 3 levels, the second has 2 levels. Okay, and a certain number of our participants will be in each cell of this design. So for an independent groups design, there'd be a certain number of participants who are in condition 1 of independent variable 1 and condition A of independent variable 2. So we could and each of these cells of that table, the kind of crossing of IV1 level 1 with IV2 level A, IV2 level 1 with IV2 level B, we would have a number of participants in each cell. Okay, and you could capture certain information in a table like this. So for a repeated measures design, we could use the same sort of table. It would be the same sort of thing, but it would be the same participants in each cell, just at different times. So if IV2 was still a between subjects factor, and IV1 was repeated measures factor, that would make it a mixed design, and levels 1, 2, or 3 of IV1 would involve the same participants, just at time 1, 2, and 3, essentially. Okay, so in a table like this, you obviously wouldn't put all the participants' score. Certainly if you had a large number of participants in each of the cells, but you could use this to kind of in a tabular form, record the ends, the number of participants in each group, in each condition, and for this particular table, that would be 6 different cells, 6 different conditions. Okay, and you could also use a table like this to record summary statistics, such as the mean for each group. So the mean score for those in group A1, and the mean score for all those participants in group A2, A3, B1, B2, B3, etc. Okay, so what we're going to do this week is focus on factorial designs with 2 independent variables, what are often referred to as two-way designs, and each of those independent variables will have either 2 or 3 levels. Okay, a word of caution in relation to factorial designs such as this: a factorial design with more than 3 independent variables becomes very hard to interpret. So I wouldn't recommend it. Usually you will find that people use 2 and at the most 3 when they're doing experiments of this type where you've got multiple categorical variables. If you have more than 3, actually interpreting what is going on becomes quite tricky. But a two-way ANOVA that we're going to look at, so we've just got 2 independent variables, allows us to test 3 hypotheses. The first hypothesis is what is the main effect of independent variable 1. The second is what is the main effect of independent variable 2. And the third is there an interaction between independent variable 1 and independent variable 2. And these are what we're talking about here is with respect to scores on the dependent variable. Okay, so let's unpack this a little bit more. So we've got our two-way ANOVA, we've got two independent variables. So a main effect refers to the effect of one independent variable or factor summed or averaged over all levels of the other independent variable. Okay, so another way to say this would be, in isolation, how does each independent variable affect scores on the DV? Are the group means different for independent 1 and for independent variable 2 separately? So it's kind of a main effect would be like ignoring the second independent variable and just looking at the effect of the first independent variable, which is kind of similar to a one-way ANOVA design. And when we look at post-hoc testing or tests of simple effects, that will become a bit more apparent, I think. Secondly, their interaction. So this is when the effect of one independent variable, so one of our factors, is not constant across all levels of the other independent variable. Okay, so in combination, we could say, does the effect of one independent variable change depending on which level of the other independent variable that we're looking at? Okay. So just briefly to sort of build up the idea about this. So a couple of weeks ago when we did ANOVA, I did some drawing and I showed you guys how we could work out the total sums of squares by fitting the grand mean, and then we could separate that into the sums of the group, some squares for the group, or sum squares between, as it's referred to on this slide, and the sum of squares error, or the sum of squares within. And those, the total sum of squares, when we're looking at deviations from the mean, can be separated into the distance of the group means from the grand mean, and then the scatter around the group means, so how much error there is, how much spread there is around each of the group means. Okay, so that's what the partitioning of sum squares looks like for one-way ANOVA, which was what we've previously covered. For a two-way ANOVA, we can split this even further. Well, the thing we're splitting further is the sum of squares for the groups, so the difference between the groups and the grand mean, and that can be separated into, or partitioned further into, the sum of squares group split into the variability accounted for by the main effect of the first independent variable, what's referred to on this slide as sum of squares F1, sum of squares for factor 1, the main effect of independent variable 2, or the sum of squares for factor 2 on this slide, and the interaction between the two independent variables, so the sum of squares for factor 1 and times factor 2, as it's shown on here. All right. So when we run an ANOVA, what we get here is a SPSS ANOVA table output for a two-way factorial ANOVA in SPSS, and it gives us what we get is 3 F ratios as shown here. One for the main effect of independent variable 1, which I've highlighted in red, one for the main effect of independent variable 2, which are effectively F ratios of ignoring the presence of another independent variable, and one for the interaction between those independent variables. So in green there, the interaction between IV1 and IV2. So on this is kind of a made up example, and I've called them IV1 and IV2, and I've called the DV for illustration. But on this data set, which is a 2x2 ANOVA, so we've got from the data set that I used, we've got 2 independent variables, and each of which has 2 levels. Yeah, you can see you can see highlighted in red. If you look at the F value and the significance, you can see we have a significant F ratio for the main effect of independent variable 1 for IV1, a significant main effect for IV2, and a significant interaction And what I'll do shortly is cover how we make sense of these types of results. Okay, when we get significant main effects and interactions, what it all means. But first of all, let's look at some plots for the different group means for each of these effects. Okay, so this plot here shows the main effect of independent variable 1. And it has two levels, condition 1 and condition 2, I've called them. And we can plot the mean for both conditions with respect to the DV. And the line is just there to help with the visualization. So the blue dot on the condition 1, which is just kind of between, it's about 5.1 or just under 5.1. That's the mean score for condition 1 on IV1 with respect to whatever the mean was, the dependent variable, with respect to, whatever the dependent variable was, score on the dependent variable. And for condition 2, this blue dot is just above 6.2, so maybe 6.25, something like that. And what that is showing is it's the mean scores for that group for condition 2 for IV1. The line drawn between them is just kind of so you can see how different they are. There are no data points along that line. It's just kind of to help us visualize. Okay. And we know from the SPS output that the difference between groups, or the F ratio, was significant. So this is the data that was on the slide here. And we know that for the main effect of IV1 was significant. So we know there is a significant difference between condition 1 and condition 2. And because we've only got two conditions, we can tell just by looking at this plot and looking at a table of descriptive statistics, so looking at the means of the two conditions, that condition 2 has a significantly higher mean than condition 1. So as you can see there, this is when we're looking at a main effect, we're kind of looking at how it interacts with the DV, or how it affects scores on the DV, whilst ignoring any other categorical independent variable. Okay, likewise, I'll do that again just so you can see the slide does change. If you look, this is IV2. If you see at the bottom on the x-axis, it's labelled IV2, and I've labelled the two conditions, two levels, level 1 and level 2, just so it was clear that this is a difference, just to differentiate it really from IV1, which is called condition 1 and condition 2. We know, so this is the same as the previous plot. It's a plot of the main effect of IV2 with respect to the DV. The blue dots represent the group means. The line between them is just kind of so it looks nice, and it is nice for us to visualize. And we know from our ANOVA table that the main effect of IV2 was also significant. And we can see here that level 2 is significantly higher than level 1. That's what that tells us, essentially. In fact, we've got a significant difference. And likewise, because we've only got two groups, we can just look at the means and say, okay, significantly different means, level 2 is significantly higher than level 1. Okay. So for both of these main effects, hopefully it's clear we're kind of looking at the independent variable in isolation with respect to their scores on the dependent variable. So it's very similar to if we were just conducting a one-way ANOVA. Okay, so this is the interaction plot. So this is a plot of the interaction. And this shows all of our groups, or all cells in this particular design. Remember, it was a 2x2, so IV1 has two conditions, IV2 has two levels. So altogether we have four groups, so we've got four means plotted here. And again, the lines are just nice for illustrative purposes. So the points at the end of each line represent a group mean. So the four groups we've got, the four cells we've got, are we've got a mean for the participants who are in condition 1 of independent variable 1 and level 1 of independent variable 2. Also, we've got those who are in condition 1 of independent variable 1 and level 2 of independent variable 2. The third one would be those in condition 2 of independent variable 1 and those in level 1 of independent variable 2. And the fourth group there would be those in condition 2 of independent variable 1 and level 2 of independent variable 2. Okay, so recall from the SPSS output a few slides back, we also had a significant interaction. So the F value in relation to the interaction, which was shown as IV1 times IV2, that was significant. And notice here that different things are happening at different levels of each independent variable. If we recall that the significant main effect of IV1, for that, scores in condition 2 were significantly higher than scores in condition 1. But if we include IV2 in the model, we see that scores in condition 2 are only higher than scores in condition 1 at level 1 of IV2. Okay, so the green line there essentially is significantly higher than the blue, or the green data point for IV2 level 1 is significantly higher than the blue data point, which represents condition 1 in participants who are also in level 1 of IV2. Okay. In contrast, at level 2, if we look at level 2 on the x-axis there for IV2, the scores are about the same, or the pattern is actually reversed. You can see there that the blue data point for level 2 of IV2 is higher than the green data point, which they're not that much different, so it's unlikely there's a significant difference between them, but the lines cross and the kind of pattern of scores is reversed. So when a participant is in level 2 of IV2, then they score the same as, or potentially higher, if they're in condition 1 than if they're in condition 2. And this type of interaction, where the lines cross, where the visual depiction of it, where there's a different pattern of means depending on which of the groups is crossed with which other group. This type of interaction, and if we draw the lines on it, shows them crossing, is called a disorderly interaction. Okay, and it has implications for what you can conclude from the results. Okay, because whether someone scores higher in condition 2 for IV1 than condition 1 depends on which group they're in for IV2, if it's level 1 or level 2. Okay, so a lot of text on this slide, I apologise, but let's elaborate a little bit further. So a significant interaction means that the additive effect of one x variable on the y variable is different at the different levels of another x variable. Okay, so we saw that on that previous slide. The means, there's a different pattern of means depending on which level of IV2 you're in. Okay. When interactions are not significant, when we're interpreting the results of an ANOVA, when we don't have a significant interaction, there are two simple stories that we can tell from the analysis, and that involves interpreting each of the main effects. They might both be significant, or only one might be significant, or neither. But when we don't have a significant interaction, the thing we focus on is any significant main effects. Okay. If the interaction is significant, though, there is one much more complicated story, and that involves interpreting the interaction. So it involves trying to pick apart what's happening differently at different levels across the independent variables. Okay, and additivity just refers to it, additivity as it's stated in that first bullet point on this slide. I thought I'd elaborate on what is meant by that to avoid. So it says a significant interaction means the additive effect of one X variable on the Y variable is different at different levels of the other. And what I mean by additive effect is that interactions test for additivity. If the model doesn't include a significant interaction, we're assuming the variables combine together additively. Okay? A significant interaction means it's not appropriate to assume that our independent variables have an additive effect on the dependent variable. So what this means is if we have a pattern of means where group 2 has a higher mean than group 1 on one independent variable, if we added a second independent variable and there wasn't a significant interaction with them, it might increase or change the scores in some way, but it'd always be the case that group 2 had a higher mean than group 1. Okay. So let's talk a bit more about that. So I mentioned previously that, if I go back 2 slides, this is what's known as a disorderly interaction. The lines on the interaction plot cross over. Okay, so we can classify interactions in different ways, and I found this very useful figure in a multivariate stats textbook, which will hopefully explain how we classify interactions. And what we've got on in these images is a 3x3 design. They're interaction plots for a 3x3 design. Okay, so on the x-axis, you've got 3 groups that relate to colour: red, blue, and green, and the lines represent the second independent variable, and there are 3 levels for that as well: balls, cubes, and stars. So we've effectively got 9 group means that are being compared here: red balls, red cubes, red stars; blue balls, blue cubes, blue stars; green balls, green cubes, green stars. I'm not sure what I think it's just an illustrative example. It's not. I'm not sure what sort of research it might relate to, if any. Okay, so when we have no interaction, then the effect of each treatment, that's the differences between groups, they're constant at each level, and the lines will be roughly parallel. Okay, so you can see in that top plot there that relates to that it's an interaction plot where there is no interaction between the independent variables. And you can see, although the means are different for different groups, so the green mean is higher than the blue mean, is higher than the red mean, for all 3, for stars, for cubes, and for balls, they differ, but the lines remain roughly parallel. And if you learn about factorial designs at undergraduate level, you will likely shown a plot like this and told when the lines are parallel, there's no interaction. And that's what this is showing, just with a 3x3 design. Okay. The second image there, the one in the middle, relates to what's called an ordinal interaction. Okay. And here you have a significant interaction. The lines are not parallel. You can see that the lines they're not even roughly parallel. You can see the lines, they get much closer together as you move, as you compare the different levels of shapes for the blue group compared to the red group, and then closer together again for the green group compared to the blue groups. Okay, so they're not parallel, but the lines do not cross. They're not crossing over. And the effects of an independent variable are not the same across all levels of the other independent variable, but the group difference is always in the same direction. You have a significant interaction and the main effects are constant. So you would report and interpret the main effects and the interaction. Okay, so if you can see here that there is a significant interaction, there are different things happening at the different levels of colour, but it's always the case that balls, that top, in terms of shapes, that top finely dotted line, the mean is always higher than it is for cubes or it is for stars. Okay. All right, and it's unlikely at undergraduate level that you would talk about ordinal interactions like this. The third one there is the disorderly interaction. Okay, and this was the type we saw a couple of slides back for that 2x2 design. And here the differences between levels switch depending on how they are combined with levels of the other independent variable. Okay, in this case, main effects cannot be interpreted because they do not represent a consistent effect. Okay, and it's likely that the type of interaction you were taught about at undergraduate level is likely to have been a disorderly interaction. It's sometimes referred to, some people refer to it as a crossover interaction. Okay, and you can see even if you have a significant main effect, it becomes meaningless because a main effect is saying this group has a higher mean than that group. So you know, in the no interaction one at the top, the main effect there would show that balls always has a higher mean than cubes, which has a higher mean than stars. Yeah. In this case, balls has a higher mean than cubes. In the disorderly case, balls has a higher mean than cubes, which has a higher mean than stars, but not if you're in the third group, which is green. Okay, it's only in the red and blue, so you can't validly interpret the main effect because which group has the higher mean depends on the other independent variable that they've been crossed with. Okay, so interpreting main effects. When your interaction is not significant and your main effects are, then you can just interpret the main effects. Okay, that's the two simple stories. Yeah, one or both could be significant. In this case, you can either look at descriptives to if the main effect only has two levels, then you can look at the main scores and know which group is higher. So in that particular case, you can just look at descriptives to interpret the results if you've got two levels. Or if your main effect has more than two levels, then you should do post hoc tests. Remember, we need to do those follow-on tests to see those multiple comparisons, those pairwise comparisons, using some sort of correction, such as Tukey, SNK, Bonferroni, whichever is your preference, or even a robust one if needs be. Interpreting main effects and interactions, the complication comes in, as we just saw on that slide, with the three types of interaction. The complication comes in when both the interaction and one or more of the main effects are significant, because the main effect may only be significant due to the interaction. So it's reaching some level of significance due to the interaction, but it's not a general trend. What the trend the main effect is showing isn't consistent, it isn't general, because it changes when you throw the other independent variable into the mix. Okay, so if we have an ordinal interaction, then we need to do post hoc tests for the main effects, and you should do some tests of what are known as simple effects for the interaction. So simple effects is just the name used for post hoc testing on an interaction. If you have a disorderly interaction, tests of simple effects are necessary. You have to conduct them to understand the interaction. You can, and when if you're reporting a disorderly interaction, even if you had a significant main effect, you could state in your results that the main effect was significant but not consistent due to a significant disorderly interaction. Okay, so I think it's probably a useful thing to do would be kind of just to walk through an example to sort of make this a bit more concrete rather than abstract. We'll obviously work through and run these types of analyses and look at interpreting the results in our lab classes this week, but let's consider an example like this. So we've got a table with our two independent variables shown on there. Factor 1 is drug, Factor 2 is type of diagnosis for some sort of mental illness. So drug, there are three types of drug: drug A, drug B, drug C. And the diagnosis there is our participants are either schizophrenic or depressive. So they've got either schizophrenia or depression. Okay, and this is data that someone's made up, and there are only two participants in each of the groups in each of the cells, and their actual scores are given there. So for schizophrenics who take drug A, the first participant got a score of 2, the second got a score of 4. For depressed people who took drug A, first participant score of 1, the second a score of 1. And the DV, those scores for the DV relate to a mean improvement for a given drug treatment. Okay, so two people with schizophrenia were given drug A. The first showed a mean improvement score of 2, the second a mean improvement score of 4. Two people with schizophrenia were given drug B. The first showed a mean improvement score of 5, the second a mean improvement score of 7, etc. Two people with depression were given drug A, and the first showed a mean improvement score of 1, the second showed a mean improvement score of 1. And you can see in these cells there's some additional information that's been made available. So the mean for each group, X bar, is shown there. So for the first cell, schizophrenics who took drug A, the mean is 3. Schizophrenics who took drug B, the mean is 6. X bar equals 6 in that case. And at the end of each of the rows, we've got the marginal means. So we've got the mean X bar for each of our type of diagnosis or type of medical problem. And you can see there for schizophrenics, the mean improvement was 4, and for depressives, if you look at X bar, the final column, the mean improvement score was 2. And the way that's worked out is quite simple. Hopefully you can figure out where I'm getting those numbers for. If we take the mean for schizophrenics for group A, which is 3, the mean for schizophrenics who took drug B, which is 6, the mean for schizophrenics who took drug C, which is 3. If we take those three, add them together, divide by 3, you'll find we get a mean of 4. Likewise, for depressives, we have got an overall total improvement score of 2 across all 3 drugs. If we look down the columns, then we've got X bar at the bottom of each. So we've got column A there, column B, column C relating to it. Then we've got X bar of 2, and that is the mean improvement score for every person who took drug A. So schizophrenics and depressives. So that's, if you look at the mean for column A for schizophrenics, it's 3. The mean for column A for depressives, it's 1. 3 plus 1 is 4, divided by 2 gives you a mean of 2. And the mean for drug B overall is 4. So, and the mean for drug C overall is 3, the bottom one. And then in this very final bottom right hand cell, we've got an X with 2 bars over the top of it, so that's the mean of means. So that's taking these marginal means, of which there are 5 cells, and working out the mean for each of them. Hopefully you can figure that out. That's kind of 2 plus 4 plus 3 for the marginal means for drug A, B and C, and 4 plus 2 the marginal means for schizophrenia and depressives. Adding them together and divided by 5 gives you a mean of means of 3. The interesting thing about these the X bar column and row, the figures in there, what I'm referring to as the marginal means. So if we look at the X bar column, we've got 1 that's 4, 1 that's 2. These are effectively the main effects. And the same for the X bar row. These are the main effects, effectively. The marginal means. They are the improvement scores for schizophrenics and depressives, regardless of which drug they took, or the improvement scores for drug A, B and C, regardless of which type of problem they are. Okay, so a lot of information packed into a small slide. Okay, so this is an interaction plot of that set of data there, and you can see the lines are not parallel. There's something different. Schizophrenics show a higher mean improvement on drug A than depressives. The same on drug B than depressives. But about the same, well, exactly the same, because the means were both 3, drug C. So we've got a significant interaction. I guess the question is it ordinal or disordinal? Okay. So in order to interpret any potential main effects, an analysis of simple effects should be conducted. Okay. A simple effect is the effect of one IV at a particular level of the other independent variable. Okay, so this is taking an interaction plot like that. And there isn't a crossover here, but I've chosen sort of an edge case where we've got potentially significant differences on 2, but not on 2 of the drugs, but not on a third drug. So potentially significant differences on drug A and B, but not on drug C. Okay, so that would mean that we couldn't really interpret the main effects. But what we're going to do is a suite of simple effects, yeah. And for this particular example, so remember, simple effects is it's basically multiple comparisons, where we compare different levels on one IV at each whilst holding the levels of the other independent variable constant. So for example, there are 2 simple effects for type of drug and 3 simple effects for type of problem. And in order for a main effect to be interpretable, the simple effects for that variable must be the same for all levels of the other independent variable. Okay. So there are 2 simple effects for the type of drug. The effect of drug for the schizophrenics and the effect of drug for the depressives. So the effect of drug, to test the first one, the effect of drug for schizophrenics, what we would do is conduct a one-way ANOVA comparing the 3 types of drugs for schizophrenics only. Okay. For the second one, the effect of drug for depressives, we'd conduct a second one-way ANOVA, one-way independent groups ANOVA, to assess if there's any difference between the scores of the 3 drugs for depressives only. So we're only considering depressives. And if the effect of drug is the same for schizophrenics and depressives, then there's an interpretable main effect for drug. And the question here is there? And the answer is no. So the question we're actually addressing is the effect for drug consistent? Is it the same for schizophrenics and depressives? So what I've just said in a very wordy manner is probably better displayed on this particular graph. So I've highlighted, so what we're doing is we're going to just take each problem type one at a time. So we're just going to start with 3 data points, the 3 means for schizophrenics on drug type. And we're going to do a one-way ANOVA comparing the mean schizophrenic score, improvement score on drug A with the mean schizophrenic mean improvement score for schizophrenics on drug B with the mean improvement score for schizophrenics on drug C. So the things that are kind of roughly being circled there by those kind of blue oval shapes. Yeah. So that's our first one. And then we can see, and then likewise we're going to just do another one-way independent ANOVA just on depressives, and we're going to compare their mean improvement score for depressives just on drug A, mean improvement score for depressives on drug B, the mean improvement score for depressives on drug C. So an ANOVA comparing the 3 points for depressives on that blue line there. Okay, that's being kind of circled around with kind of like a greyish green type of oval shape. All right. And what we can see just from looking at this is for depressives. Remember, what we're interested in is at the bottom: is the effective drug the same? Is it consistent for schizophrenics and depressives? And the answer to that is, for depressives, drug C, the blue line, has a higher mean and is better than drug B, which has a higher mean and is better than drug A. For schizophrenics, the red line, drug B is better than both drug C and drug A, which seem to have the same means. Okay, so there isn't, in this case, with this particular data, there's no interpretable main effect, because which drug is best depends. The best drug to take depends on whether you're schizophrenic or depressive. Okay, so that was 2 one-way ANOVAs for comparing type of drug, so simple effects for type of drug, effectively what we're looking at there. The next thing we need to do is simple effects for type of problem, and there are 3 simple effects or 3 comparisons that we need to make for type of problem: the effect of type of problem for drug A, the effect of type of problem for drug B, the effect of type of problem for drug C. So what we could do to address the first one is conduct a one-way independent groups ANOVA or t-test, because we're only comparing 2 means, to assess if there is any difference between the scores of the participants who took drug A only. Okay. For the second one, similarly, conduct a one-way independent groups ANOVA or t-test to assess if there's any difference between the scores of participants for drug B only. And then thirdly, the effect of type of problem for drug C. We're going to look at conducting a one-way ANOVA or t-test to assess if there's any difference between the scores for the participants who took drug C. Okay, so effectively 3 tests, each comparing 2 groups, so 3 t-tests or 3 ANOVAs. Okay, if differences between schizophrenics and depressives are in the same direction for all 3 types of drug, then there's an interpretable main effect for type of problem. In this case, we're asking, is there? And the answer is, once again, no. And a point worth remembering is that because we're doing multiple testing, so if you did 3 t-tests to make these comparisons, you should use some sort of Bonferroni procedure to control for type errors. And the question we're actually addressing here, similar to the previous one, is the effect of type of problem consistent? Is it the same for all 3 types of drug? Okay, so what's that mean? It means we're doing these 3 comparisons that have been highlighted on the slide. Firstly, we're just looking at drug A and ignoring the other 2, and then we're comparing schizophrenics, the improvement score for schizophrenics, to their depressives. And what we'd likely see here is that schizophrenics significantly higher improvement score on drug A than depressives. And then we do a second test, our second t-test, say, for drug B, and we compare those 2 points that are highlighted. So in this case, again, what we would find, I'm pretty sure, is that the mean improvement score for drug B is better for schizophrenics than it's for depressives. So that's consistent with drug A as well. The next one, though, the third one, is our test that's highlighted, which is comparing the 2 points, 2 types of problem, and how they improve when taking drug C. And what we'd find there is no difference between the 2. Okay, so schizophrenics do significantly better on drug A than depressives. Schizophrenics do significantly better on drug B than depressives, but there's no difference between the 2 on drug C. So if you're schizophrenic, you want drug B, okay? Because it depends. But if you notice, drug C is the is the one that leads to the highest mean improvement score for depressive. So if you're depressive, you want drug C. Okay, so if you're schizophrenic, you definitely want to be taking drug B, get better overall improvement. If you're depressive, you definitely want to be taking drug B. That's the highest mean for all three in relation to depressives. So the answer, what we're finding here is what's the best drug to improve, what we're asking, what's the best drug to take to improve, to improve your symptoms. And the answer is it depends. It depends if you're schizophrenic or if you're depressive. Okay, so there is no crossover in this case, but it's still a disorderly interaction. Okay, the lines are not crossing, they are the same, but we can't. There is that even if we got two significant main effects, we definitely get a significant interaction, and the significant interaction in this case would make the significant main effects not interpretable, invalid essentially, because different things are happening at different levels. Okay, so that's it. That's all I'm going to cover. The references for this week, Factorial ANOVA, are chapter 14 of the Andy Field textbook, and I've also included again Tabachnik and Fidel's Using Multivariate Statistics textbook. It's a much more difficult textbook, but it's pretty good, and chapter 7 of that, I think the most recent version, chapter 7, covers Factorial ANOVA. Okay, so this is Factorial ANOVA, quite a complex thing to cover in a lecture. The lab classes, when we run these in lab classes and look at interpreting the results where we've got actual data and figures, should make it a little bit easy to understand. But all we've done in these last few slides here is we've looked at this data, this example data. We've seen that there's, if we look at the plot, we can see straight away there's going to be some sort of interaction. We're not entirely sure what's going on, so we've conducted simple effects tests where we've done effectively 5 comparisons. We've conducted a one-way ANOVA holding type of problem constant and comparing which of the 3 drugs are better. For schizophrenics, we've done another one-way ANOVA holding type of problem constant and looked at the effect of the different drugs just for depressives. We've then done 3 more tests of simple effects where we've basically compared, we've held type of drug constant, and we've looked to see if there's a significant difference between the two diagnoses in terms of the mean improvement score. So we've done a t-test for drug A comparing schizophrenics with depressives, a t-test for drug B comparing schizophrenics with depressive, and a t-test for drug C comparing schizophrenics with depressives, all Bonferroni corrected. Okay. So that is the way we would break down a significant interaction. We get a significant interaction, we've then got to pick it apart by doing these multiple comparisons. Right, I'm going to stop talking. Hopefully that is useful, and I will see you soon.